Hello everyone. In last video, we discussed in detail about the different type of changes that occur in the cell during anaphase-I. Now in this video, I am going to focus on last stage of karyokinesis 1 of meiosis-I, that is telophase-I. Okay, telo is end and phase is stage. So this is the stage which end the process of karyokinesis. After this stage, the cell will going to have two daughter nucleus. Okay, so we will going to see all the type of changes that occurred during the telophase-I in typical eukaryotic cell like animal cell or plant cell. First thing happen during the telophase-I is dissociation and disappearance of the spindle fibers. Okay, we know that there are two important type of spindle fibers arise from the centriole like asterase and actual spindle fibers, but in the telophase now there is no need of these threads or fibers, so a centriole will going to dissociate from the spindle fibers and now the spindle fiber get disappear. Okay, only we can see is the centriole at two pole of the cell. Now in the telophase, the separated chromosome from the pair get reach to the respective pole. So now this centriole will going to have two chromosome initially. Both the chromosome have exchanged genetic material, that is fragment of opposite chromosome. Same case with the opposite pole. Here you can see the movement of the chromosome completed, they reach to the respective poles. Okay, two chromosome get stabilized to one end while remaining two get stabilized to the opposite end during the telophase. The nuclear membrane and nucleolus which is disappear during the prophase-I again reappear during the telophase-I. Okay, so now the cell will going to have nucleus with nuclear membrane and small body which is present in the nucleus, that is nucleolus at both the pole. So this is nuclear membrane and nucleus have nucleolus. So nuclear membrane and nucleolus reappear during the telophase-I. After reappearance of the structures, the chromosome which is present inside the nucleus now start decondensation process. We know that during the prophase-I, condensation occur, water molecule come out of the chromatin and chromatin get converted into thick short structure chromosome. Exactly opposite process happen during the telophase-I. Due to decondensation process, water molecule is absorbed by this chromosome and this short thick structure converted into thin and long chromatin. So now chromosomes are no longer visible inside the nucleus. Instead of chromosome, we can see a network of thin, long chromatins inside the nucleus. Okay, so three important things which occur during the telophase-I is reappearance of the nuclear membrane and nucleolus, disappearance of spindle fibers, and chromosome get converted into thin and long structures called chromatin. So these are the chromatins. When we start discussion of the karyokinesis-I, we have take the example of cell which have 4 chromosomes in every nucleus. Now in this case you can see this cell have two nucleus and each nucleus only have two chromosomes. That means genetic material get reduced. But remember that the the nucleus having n number of chromosome, that is nucleus is haploid, but the DNA content C value is 2C, because initially we have take the example of cell in which uh, the cell is diploid and DNA value is 4C. So out of these two diploid n, one n is present in each nucleus. So this nucleus will going to have one n and this nucleus will going to have one n chromosome. And out of this 4C DNA, 2C DNA or half DNA is present in this nucleus and remaining half is present in opposite nucleus. So this is the condition at the end of telophase-I. All the cell organelles which is already present in double inside the cell going to get their membrane in telophase-I, like endoplasmic reticulum, Golgi bodies, mitochondria, all will going to get their new membrane at the time of formation of nuclear membrane. All membrane get produced inside the cell. The cell will going to have double amount of cytoplasm in it. And now this cell is ready to enter in last stage of meiosis-I, that is cytokinesis-I, the process in which cytoplasm of the cell get divided, and during the cytokinesis-I, the one cell get divided to produce two daughter cell. In my next video, I am going to discuss in detail about the cytokinesis-I of meiosis-I. Thank you.